Hi class, welcome to our lesson in General Physics 1. This is Sir Alex Vasco and today we'll be discussing meiosis. Last time we've discussed about mitosis, which is the cell division for somatic cells. In lesson 5.2 we'll be discussing meiosis, which is cell division of sex cells. The goals of today's lesson will be for us to be able to describe the stages of meiosis. Similar with mitosis, meiosis has its own stages. Next, for us to be able to explain the significance and or the application of meiosis. So let's begin by discussing what is meiosis. Basically, meiosis, like I mentioned earlier, it's the way for sex cells to produce, to reproduce, and They do this by way of cell division. How is it different though from mitosis? Mitosis are done by the somatic cells, meaning most of the cells in our body undergo mitosis. But meiosis is more specific for sex cells or gametes. That is because in mitosis, we create two daughter cells that has the same sets of chromosomes, meaning it starts from 2N parent cells, and when the two daughter cells were created, those two daughter cells also contains 2N, meaning they also contain the two sets of chromosomes required for the cell to function properly. However, for sex cells, or in case of human, egg cells and sperm cells, the four daughter cells that are there are not only two daughter cells were created, there are actually four daughter cells that are created. Moreover, they have the half of the number of chromosomes in each of the daughter cells. So in mitosis, we create two daughter cells that are 2N. But in meiosis, we want to create four daughter cells that are 1N. So meaning there's only one set of chromosome available in those gametes. And this is essential in order for us humans to create new human being. Imagine if our sex cells are already at 2N. What happens is that when those sex cells or when the sperm cell fertilizes the egg cell, the human created would have 4N, and that is ridiculous because that zygote will not survive because there's too many chromosomes inside the cell. So in order for us to create a normal human being, we have to separate the sex cells that will fertilize into human being. We have to create them into 1N. So they only have to have one set of chromosomes. And today we'll be discussing how one parent cell of 2N, it starts with 2N, this meiosis, ends up into creating 1N of daughter cells. For 1N exactly, for 1N daughter cells. Okay, so these happens via phases. So in meiosis, we actually undergo two division, two process of division. We have meiosis 1 and meiosis 2. Meiosis 1 includes prophase 1, metaphase 1, anaphase 1, and telophase 1. In meiosis 1, we actually separate homologous pairs. Homologous pairs, we'll discuss what are those later, but it's basically the partner of a pair of chromosomes. In meiosis 2, we have prophase 2, metaphase 2, anaphase 2, and telophase 2. In meiosis 2, we actually separate sister chromatids. So in mitosis, when the somatic cells divide, what is being divided are actually sister chromatids. But for us in meiosis 1, we have to separate first the homologous pairs. In meiosis 2, that's when we separate the sister chromatids. So let's discuss what is happening in meiosis 1. Similar with mitosis, meiosis 1 has its own PMET, prophase, metaphase, anaphase, and telophase. So those are the stages of meiosis. But in this case, since this is the first part of meiosis 1, we'll name those as P1, prophase 1, metaphase 1, anaphase 1, and telophase 1. In meiosis 1, before the cell enters meiosis 1, the cell has to go through interphase. So the interphase in sex cells are actually basically the same with mitosis. It's the same process in G1 wherein the cell doubles its size or it increases physically, and that is because everything is being duplicated inside in preparation for separation. In S phase, the cells are also, the DNA inside the nucleus is synthesized or duplicated. That's the same thing that is happened before the sex cells enters meiosis 1. And same too, it's the same thing. It's a stage of preparation for meiosis 1. So the interface for sex cells is actually not different for the interface in the mitotic cells that we've discussed previously. So after undergoing interphase, the cell enters meiosis 1 by first entering prophase 1. Most of what is happening in meiosis 1 actually happens, or some of the most important things that happen in meiosis 1 actually already happens in prophase 1. What happens first is, similarly with mitosis, the chromosomes condenses. But moreover, homologous pairs partner up. So what is homologous pair? So let's kind of like draw what is homologous pair. You know that a cell contains two N. So one cell contains two N. And this means that there are two pair of chromosomes. We know that the human cell contains 46 chromosomes. And these 46 chromosomes, half of them comes from the father, and the other half comes from the mother. So meaning 23 comes from the father, and the 23 comes from the mother. So this is what we call chromosomes, and there's 46 of them, 23 from the mother, 23 from the father. That's what is partnering up. So the chromosome pair, so if we draw, for example, this is the chromosome that comes from, so this is the chromosomes that comes from the father. So imagine this is already 23 on this side. And then we have the chromosomes that comes from the mother. So they actually have to pair up. And in men, we actually have one from X and one Y, which always comes from the father. So let's just erase this for now. So we have a Y that specifically comes from the father, and then we have the X that comes from the mother. For women, women usually has excess, not usually has two X's, and that can either that come from both the mother and the father. So imagine this line as the chromosome that come from the father, and the red chromosomes are the chromosomes that come from the mother. So these pairs actually line up. And in meiosis 1, that's actually what we want to separate. In the end, in meiosis 1, we are actually creating already one N of, or it means the cell that is created after meiosis 1, there's only one set of chromosomes present inside the cells already. And that results because of the pairing up of these homologous pairs. So, moreover, these homologous pairs are actually connected by what we call synaptonemal complex. So you can think of this purple right here as coming from the mother. Let me just change this. So think of this pinkish color as coming from the mother, and the purple bill at the back as the chromosome from the father. So these two partner up together and they're held together by what we call synaptonymal complex. Moreover, what is happening during prophase 1 in meiosis 1 is that there's some crossing over happening. So what that means is that these pair of this homologs, this pair of chromosomes, actually switches or exchange genetic materials. As you can see here, this is one chromosome and this is the pair chromosome. This is the homolog. We call it the homolog. And these A, B's, and C's are basically genes. And they code for the same thing. So this gene A here codes, and this small letter A gene here, they both code for the same thing. So what they can do is they can actually switch those genes. So as an example here, the C genes or the gene C from one of the homologs switches with the C gene of the other homolog. This is what we call crossing over, and this is very important for diversity. That's why no human being is actually the same, of course, aside if you are maternal or paternal twins. But for most of humans, most of your genes are actually very different because of crossing over. There's some, so you can see here in this moving picture right here, the switches. So that's what we call crossing over. And then at the end of prophase 1, it ends when the centrosomes it starts to move apart, and then the spindles, if you guys remember, the spindles are the microtubule spindles. They start to form and slowly start to attach onto the chromosomes. Okay, so for prophase 1, just a summary, this is what is happening. So the DNA condenses after it is replicated in S phase during interphase. And moreover, not only does it condense, the homologs starts to pair up. So the homolog pair from the mother and the homolog pair from the father, they start to pair up. And they are connected together by what we call the synaptonymal complex. Moreover, after pairing up, these chromosomes actually switch genes. We're not going to discuss anymore how they do that because that's quite a lot of process and quite complex on how they do it. But you can think of it as one gene and the allele or the gene that basically codes for the same thing, they switches, which is what we call a crossover. And then prophase 1 ends when the centrosomes starts to move and the spindles starts to form to start attaching onto those chromosomes. When this happens, we move to metaphase 1. In metaphase 1, the spindles capture the chromosomes and move them in the center of the cell called metaphase plate. Each chromosome is only attached to one side of the cell, unlike in mitosis where there is no pairing up of the homologs. So you basically have those 46 chromosomes. You have those 46 chromosomes lined up in the metaphase plate. So they line up individually. But for the meiosis, in the chromosomes, you only have the 23 lined up in the metaphase plate. So how can I, what that means, let's see. Let's kind of like draw what's happening. Okay. So in mitosis, what is happening is you have all those 23 pairs or those 46 chromosomes, so they line up individually. In meiosis, what is lining up, because these are the homolog pairs. So I hope that is kind of clear what is happening. So you have all the 46 individual lining up in the metaphase plate in mitosis. But for meiosis, these paired up, so you basically only have 23. Uh, right now you have the 23 pairs the 23 pairs lining up in meiosis. I hope that's kind of clear what is happening. That is very different in meiosis from mitosis. And moreover, this is very important, each chromosome is only attached to one side of the cell. Remember in mitosis when we have centrosomes, let's say these are centrosomes, these centrosomes when they attach, it means a single chromosome can be attached to one or it's attached to both the chromosome. But for meiosis, the centrosomes, only one of the chromosomes is actually attached to one of the centrosomes. So this all the chromosome on this side will get attached on the centrosomes on this side, and all the chromosomes at this side on the right side are only attached on the centrosomes on the right side. Okay, so only one, each chromosome is only attached to one side. So that's another difference in metaphase compared to mitosis. In mitosis, all chromosomes, all those 46 chromosomes are attached to both centrosomes. But for meiosis, since we have pairing up of homologous pairs, only one chromosome is attached to one side. And the orientation is random. So what do we mean by that? I mentioned earlier we have sets from our mother and we have sets from our father, right? Let's change the color of the father. So we have set from mother and we have set from the mother. So you can imagine this as those sets. So these are pairs. What is happening when we say orientation is random is that these pairing right here, one side is not purely from the mother and the other side is purely from the father. What is actually happening is these pairing right here is random. It could be one side could all, you could have three fathers and then the other three is on the other side, and then so it's sort of something like this. And then the mother side is also randomly placed. Some are placed on one side and the other is placed on the other side. So these red colors are from the mother. So these red chromosomes are from the mother and the black are from the father chromosomes. So when they line up, they are actually random. So when you start pulling them apart, some of the chromosomes that come from the father and some that come from the mother go to one side, and the other also receives some from the father and some from the mother. So that is what we call what is happening during metaphase plate. So after they paired up, the pairing up is the orientation of the pairing up is random, and that also contributes to genetic diversity. So as you can see here, when they phase, so that's why you have a lot of core, because this orientation of where they phase, where the mother chromosome from the mother and where the chromosome from the father phase is random, and you ended up having very different combination of this pairing up. And like I said, that contributes to the genetic diversity of the gametes you create, which means also it contributes to the genetic diversity of the human created. Okay, so like here the homologous pair line up, and as you can see here, the orientation of these pairing is random. This could have been the same way that this side is. The purple could be on this side instead, and the pink was on that side. Okay, so I hope that is clear on what is happening in metaphase. So just a summary in metaphase 1. Very similarly, after the centrosomes attach onto the chromosomes, they line up on the metaphase plate. But since we are lining up homologous pair, only one chromosome is attached on the one side, unlike in mitosis. And the orientation of these homologous pairs is random. Next, let's move to anaphase. So in anaphase, the homolog pairs are pulled apart and they move apart to the opposite ends of the cell. So you can think of this as the blue one is the father, and you can see the homolog pairs are being pulled apart. So this one whole cell is currently in a state of 2n. And then in that 2n are all paired up. So we have one n on one side and one n on the other side. So when anaphase it starts pulling them out or pulling them apart, we are already starting to create one n cells. So in meiosis 1, we are dividing homologous pairs and we are already dividing that 2n. Okay, so that one set of chromosomes already go on this side. So we have 46 originally, and that 46 is lined and up. So 23 is already coming on this side, and the 23 is already going on the other side. So that's what's happening in anaphase 1. And in anaphase 2 or in telophase 1, it's basically where we also we finally separate the cell, creating two daughter cells that has half the set of chromosomes from the original parent cell. So the chromosomes arrive at the opposite poles of the cell. In some cells, they recreate those nuclear membrane, the nucleus. The membrane of the nucleus is free-form, and the cell or the DNA decondenses. But some cells escapes the process because some cells immediately go to meiosis 1 or meiosis 2. Moreover, in telophase 1, cytokinesis also ends. If you guys remember, cytokinesis is basically the separation of the cytosol. It usually starts in anaphase or it starts in either anaphase and telophase, but it always ends after the telophase. So cytokinesis at this point has also started either in anaphase or telophase, and then it ends during telophase. We are finally creating those two daughter cells. So from here on out, the cell could enter G1 phase but they will never duplicate because we already duplicated this, remember, on the beginning of interphase. So we've already duplicated this. So this is actually, they already have sister chromatids in them. And that's what we have to separate now for meiosis 2. This sister chromatids is what we actually separated during mitosis. So now we're basically entering a mitosis stage or stage that is similar to mitosis, but the cell is already 1n, not 2n. Okay, so we already created haploid cells, or the 1n cells are called haploid cells. Then we enter meiosis 2. In meiosis 2, cells move from meiosis 1 to meiosis 2 without, again, we do not copy the DNA because we already copied it at the start of interphase. So after meiosis 1, no need for further copying. And it's more shorter and simple process than meiosis 1. Cells created in meiosis 1 are the only cells that enters meiosis 2, which makes sense because how can you enter meiosis 2 if you haven't done meiosis 1? The cells may only have one pair of chromosomes, but they are still paired with sister chromatids. So if you guys remember what sister chromatid, so sister chromatids, remember, is we begin with the DNA and then it is duplicated, remember? It is duplicated. And then when when these DNA condenses, we create what we call sister chromatids. So that's why we usually draw the DNA into axis. So the sister chromatid is it's basically the same DNA but duplicated. Okay, so so the cell created now after meiosis 1 is only 1N. It means there's only 23 pairs of chromosome inside that cell created at meiosis 1. But this 23 pairs is a duplicated pair. It means the separated homolog is actually it has a sister chromatid that we have to separate to go back to being to go back to being a so this DNA right here. Okay, so creates haploid cells with non. So at the end of meiosis 2, we are creating one N, but this time the DNA inside does not have a sister chromatid. So it's a singular or non-duplicated chromosome. Okay, meiosis 2 starts with what we call prophase 2. So it's the same thing. It's the same PMAT, only we're using 2. So here starts with prophase 2. Again, it's basically the same thing. The chromosome condenses and the nuclear envelope breaks down, or the nucleus membrane breaks down if the cell ended up making them after meiosis 1. If the cell did not create any nuclear envelope and the chromosome did not condense, no need to do this. So this only happens if the chromosome decondensed in if the cell after meiosis 1 created a nucleus. And again, the centrosomes starts to move apart from, or the centrosomes has to duplicate again because one centrosome ended up on one cell and the other centrosome ended up, and that centrosome has to duplicate again, and those centrosomes have to move apart from each side of the cells. And then we enter metaphase 2, wherein the centrosomes attach to these chromosomes. So those 23 chromosomes, it's attached. This time, well, since it's a sister chromatid, so it's a it's a chromosome with a duplicated pair, and they get attached to both the chromosome, and then they ended up in the middle. So they ended up again in the metaphase plate in metaphase 2. And after ending on the metaphase plate, anaphase 2 starts to happen, wherein we finally start to pull those sister chromatids apart. In the end, so anaphase 2, we pull apart the sister chromatids, which are connected by the along the kinetochore, if you guys remember that, in their centrum here. So in telophase, it's basically uh, you basically separate those two cells. Now we're creating four daughter cells with one N each, and these N does not have a duplicated sister chromatid, so we only have one sister chromatid in each of these cells. And then finally, after telophase, the nuclear membrane reforms to house those DNA. Chromosomes decondenses and cytokinesis splits those cells apart. And in the end, we produce four haploid cells. So that is meiosis 2. Way easier than meiosis 1. In humans, so these cells in human, that's what the cells that becomes sperm and egg cells. So mixing and matching of genes. So we've discussed this earlier. All the cells produced in meiosis are haploid. I hope everyone understand that. And none of the four daughter cells are genetically identical. That is because there is a blending of the DNA, and this happens during crossing over, where the homologous pairs exchange genetic materials along their alleles. So alleles is basically the same gene of the other chromosome pair. So you remember we have a chromosome pairs, and these chromosome pairs actually duplicate for the same thing because they both contain the same DNA or DNA code for the same expression. For example, the color of the eye and the first chromosomes for the mother, for the chromosome that come from the mother, the chromosome 1 that comes from the father actually also codes for the same eye color. So, depending on which one is the dominant, that becomes the color of your eyes. But in this case, so let's say this part of this gene right here codes for the eye color, or basically gives you eye, let's just say that. And then these part right here of the cell or of the DNA gives you or codes for the eye. These can be separated or these can be switched because they code for the same thing naman. So you would end up, you no matter which sperm or egg cells you get, you're still gonna end up having the same eyes, or you're still gonna end up having eyes. So that's what's crossing over. They just switch on the same alleles or the same gene that code for the same thing. Another thing is because of the random orientation of the homologous pair. So yung parang in-explain ko kanina. So some of the chromosomes from the mother go on the left side, and some of the chromosomes from the mother go on the right side. The same thing for the father. In the end, when they are separated, you have random number of chromosomes from your father and mother on one cell, and the random number of chromosomes from both mother and father on the other cell. And nangyayari, after crossing over and random orientation ng separation, nagkakaroon ng mixing and matching of genes. And ang nangyayari in the end is you have basically an infinite variation of gametes. So yung mga nakikreate na haploid cells are genetically non-identical. Yung DNA sample na sa loob is not the same DNA sample dun sa other sperm cells or other egg cells. Random orientation alone creates 8 million different types of possible gametes. When you pair it up or lay over more, yung ginagawa ng crossing over, because crossing over not only happens on one allele, it could happen 25 on 25 different places or up to 25. So it means this X1 right here and this X1 from your father can exchange genetic materials up to 25 times. So pairing these two up together, you basically can create an infinite variation of sperm cells and egg cells. Even though they came from one daughter cells, or one parent cells, four daughter cells na create are not genetically identical from each other, or moreover, they are not identical with the parent cell, which is very different from mitosis. So that is meiosis. I hope it's clear. If you guys have any questions about meiosis, do not hesitate to message me, and I hope clear yung lesson. And yeah, if you guys have any question, just message me, and you guys have a good day po.